Welcome back. This is Education Lesson. I'm Wesley Martin, your lecturer for today in the nervous system. And honestly, this is one of the most satisfying topics in all of physiology because it connects two very different scales. On the small scale, we'll look at how a single neuron passes a message to the next one across a tiny gap. On the large scale, we'll zoom out to see how billions of neurons are organized into a brain, a spinal cord, and the nerves threading through your entire body. And then we'll look at the part that runs quietly in the background, the automatic system that controls your heartbeat, your digestion, your pupils, without you ever thinking about it. Keep that through line in mind the whole way through. Signal, then structure, then automatic control. Let's get started. Here's the roadmap for the next module. In 4 parts. First, synaptic transmission, how neurons actually talk to each other, including the different flavors of postsynaptic response and neurotransmitters involved. Second, structure and organization, the big anatomical map. Central versus peripheral, the regions of the brain, the spinal cord and reflexes. Third, support and protection, the glial cells that do the housekeeping. The blood-brain barrier, and the cerebrospinal fluid that cushions everything. And fourth, autonomic control. The sympathetic and parasympathetic divisions, their transmitters and receptors, and the adrenal medulla. Notice the logic here. We build from a single synapse outward to the whole system, and then to how that system regulates your body automatically. Each part sets up the next. Before we dive in, here's what you should be able to do by the end. Sequence the events of chemical synaptic transmission, and tell excitatory from inhibitory responses. Explain how summation, adding signals together, decides whether a neuron fires. Diagram the divisions of the nervous system and match brain regions to their job. Describe a reflex arc and trace the signal through both simple and complex reflexes. Contrast the sympathetic and parasympathetic divisions, their anatomy, chemistry, and effects, and predict what they do to a given organ. And finally, explain dual innervation, autonomic tone, and the role of the adrenal medulla. These 6 objects map directly onto the 4 parts. If you can talk through each one in your own words by the end, you've got it. Keep them in the back of your mind as we go. Electrical and chemical synapses. There are 2 ways one neuron can hand a signal to the next. The first is the electrical synapse. Here, the cells are physically joined by gap junctions, little protein channels that let ions flow straight from one cytoplasm into the other. It's incredibly fast, essentially instant, and it works in both directions. Great for synchronizing groups of cells, but relatively rare. The second, and far more common, is the chemical synapse. Now the cells don't touch, they're separated by a tiny gap called the synaptic cleft. To cross it, the signal gets converted into a chemical messenger, a neurotransmitter. This adds a brief delay, about half a millisecond, and it only goes in one direction. So why is the slower one-way option the dominant one? Because that chemical step is where the signal can be amplified, integrated, and fine-tuned. That flexibility is everything. Let's walk the chemical synapses step by step. And this sequence is worth memorizing in order. 1. An action potential arrives at the axon terminal. 2. And this is the trigger, that depolarization opens voltage-gated calcium channels, and calcium rushes in. 3. The calcium causes vesicles, little packets of neurotransmitter, to fuse with the membrane. 4. They release their neurotransmitter into the cleft. 5. The neurotransmitter drifts across and binds receptors on the next cell. And 6. Those receptors open ion channels, producing a small voltage channel change. The postsynaptic potential. The single most important link is calcium. No calcium entry, no release. Full stop. And one last thing. The signal has to be switched off, so the transmitter is cleared from the cleft by reuptake, by enzymes breaking it down, or by simply diffusing away. Excitatory versus inhibitory potentials. So what does that postsynaptic potential actually look like? It comes in two types. An excitatory postsynaptic potential, an EPSP, happens when the positive ions like sodium flow in. The inside of the cell becomes less negative. It depolarizes. That nudges the neuron toward its firing threshold. It makes firing more likely. An inhibitory postsynaptic potential, an IPSP, is the opposite. Chloride flows in, or potassium flows out, and the inside becomes more negative. It hyperpolarizes. That pushes the neuron away from threshold, less likely to fire. Here's the crucial word for both: graded. Neither one is all or nothing, and neither one alone is enough to fire the cell. They're small votes, some for, some against. What actually determines the outcome is the running total of all those votes arriving at one special spot, an axon hillock, which brings us to summation. Summation is the neuron adding up its inputs, and it happens two ways. Temporal summation is adding over time. Picture one synapse firing again and again, so fast that each little EPSP arrives before the last one has faded. They stack on top of each other and build up. Same input, hammered rapidly. Spatial summation is adding over space. Now picture many different synapses, scattered across the neuron, all firing at nearly the same moment. Their EPSPs spread inward and combine, different inputs arriving together. In real life, the neuron does both at once, and it's also subtracting the IPSPs. If, after all that adding and subtracting, the membrane at the axon hillock reaches threshold, boom, an action potential fires and the message continues. If it falls short, nothing happens. That's the fundamental decision every neuron makes, thousands of times a second. Let's meet the main players. Acetylcholine works at the neuromuscular junction, throughout the autonomic system and the brain. Glutamate is the brain's main excitatory transmitter. GABA, or GABA, is its main inhibitory one. That pair does most of the heavy lifting in the central nervous system. Glycine is another inhibitory transmitter, mostly in the spinal cord and brainstem. Then the modulators, norepinephrine, tied to arousal and fight or flight, dopamine, central to reward, motivation, and movement, and serotonin, involved in mood, sleep, and appetite. But here's the point I really want you to leave with. A neurotransmitter is not inherently excitatory or inhibitory. The effect depends on the receptor it binds. Acetylcholine excites skeletal muscle, but it slows the heart. Same molecule, opposite results. So always ask not just which transmitter, but which receptor. Now the master map. And if you learn one diagram from this module, make it this one. At the top, the whole nervous system splits in two. The central nervous system, the CNS, is the brain and spinal cord. That's the integration and command center. Everything else is the peripheral nervous system, the PNS. The PNS splits by direction of traffic. Afferent fibers carry sensory information toward the CNS. Efferent fibers carry commands away from it. Efferent then splits again into somatic, voluntary control of skeletal muscle, and autonomic, the involuntary control of glands, smooth muscle, and cardiac muscle. And the autonomic branch splits one last time into sympathetic and parasympathetic. Notice this nesting previews the rest of the module. Those last two boxes, parasympathetic and sympathetic, are exactly where we'll spend part 4. 5 regions, 5 jobs. Let's keep it clean. The cerebrum, with its wrinkled cortex, handles the higher functions: thought, voluntary movement, sensation, and language. The diencephalon sits underneath and contains two key structures: the thalamus, which is the great sensory relay station, and the hypothalamus, the master controller of homeostasis, that drives both the autonomic system and the endocrine glands. The cerebellum, tucked at the back, manages coordination, balance, posture, and fine timing of movement. The brainstem, midbrain, pons, and medulla, runs the vital reflexes that keep you alive: breathing, heart rate, blood pressure. And the limbic system handles emotion, motivation, and formation of new memories. One connection to flag: the hypothalamus is the bridge to part 4. It's where conscious brain meets autonomic control. Let's slice the spinal cord across and look at the cross-section. In the center, you'll see a butterfly, or an H, of gray matter. That's where the neuron cell bodies and synapses are. So it's the site of integration. Wrapped around it is white matter, the myelinated tracts that carry signals up and down the cord like highways. Now watch the wiring. Sensory information enters through the back, the dorsal root, and the cell bodies of those sensory neurons sit in a little bulge called the dorsal root ganglion. Motor commands leave through the front and ventral root, heading out to muscles and glands. Here's the memory hook. Dorsal is data in, ventral is motor out. Sensory in the back, motor in the front. Nail that, and the spinal cord anatomy suddenly makes sense. A reflex arc is the shortest route from stimulus to response, often without the brain involved at all. The basic path has 5 parts. A receptor detects the stimulus, a sensory neuron carries it in, the spinal cord processes it, a motor neuron carries the command out, and an effector, a muscle or gland, responds. Now, 2 versions. A monosynaptic reflex has just one synapse, sensory neuron straight to motor neuron. That's the knee-jerk stretch reflex, lightning fast, because there's nothing in between. A polysynaptic reflex adds one or more interneurons in the middle. Think of yanking your hand off a hot stove. That needs coordination across several muscles. So interneurons step in. The trade-off is simple. More synapses mean more processing and richer responses, but each one adds a tiny delay. Speed versus sophistication. Neurons get all the glory, but glial cells actually outnumber them, and they do the essential support work. 6 to know. In the CNS, astrocytes handle support and ion balance and help build the blood-brain barrier. Oligodendrocytes myelinate the axons. Microglia are the immune cells that clean up debris. And ependymial cells line the ventricles and help make cerebrospinal fluid. In the PNS, Schwann cells myelinate axons, and satellite cells support cell bodies in the ganglia. Now the blood-brain barrier. The capillaries feeding the brain are unusually selective. Their endothelial cells are sealed together by tight junctions. An astrocyte and feet wrap around them. The result: only small, lipid-soluble, or actively transported molecules get through. That protects the brain from toxins. But it's also why so many drugs can't reach it. A blessing and a real clinical headache. Cerebrospinal fluid, or CSF, is the clear liquid that surrounds and cushions the central nervous system. Follow its circuit. It's produced by the choroid plexus inside the brain's ventricles. 4 connected chambers. From there it flows out into the subarachnoid space, bathing the entire brain and spinal cord. Finally, it's reabsorbed back into the venous blood at structures called arachnoid granulations. Produce, circulate, reabsorb. A continuous cycle. And it does 3 jobs. Buoyancy. The brain effectively floats, dropping its felt weight to a fraction of its real mass, so it doesn't crush itself. Protection. The liquid layer absorbs shock when your head moves suddenly. And stability. It keeps the chemical environment around neurons constant and carries away waste. Buoyancy, protection, stability. 3 words that capture why this quiet fluid matters so much. The autonomic system has 2 branches that pull in opposite directions, and the nicknames say it all. The sympathetic division is fight or flight. It mobilizes you for action. It speeds up the heart, dilates your pupils and airways to take more in, dumps glucose into the blood for fuel, shunts blood to your skeletal muscles, and shuts down digestion because now it is not the time. The parasympathetic division is rest and digest. It conserves and restores. It slows the heart, constricts the pupils and airways back down, ramps up digestion and salivation, and promotes storing nutrients for later. The trick to learning these is to read them as pairs. Pick an organ, the heart, the pupils, the gut, and the 2 divisions almost always do opposite things to it. Learn one side and you'll basically get the other for free. The 2 divisions also look different anatomically, and this is a favorite exam topic. Both use a 2-neuron chain, a preganglionic neuron from the CNS that synapses in a ganglion onto a postganglionic neuron that reaches the target. What differs is where they start and where those ganglia sit. Sympathetic fibers leave the middle of the spinal cord, the thoracolumbar region, segments T1 through L2, and synapse early, in ganglia right beside the cord. So they have a short preganglionic fiber and a long postganglionic fiber. Parasympathetic fibers leave from the top and bottom, the craniosacral region, certain cranial nerves plus sacral segments, and synapse late, and ganglia near or inside the target organ. So the reverse, long preganglionic, short postganglionic. Thoracolumbar, and short then long for sympathetic. Craniosacral, and long then short for parasympathetic. Now the chemistry of those pathways. There's a clean rule here. Every preganglionic neuron in both divisions releases acetylcholine into nicotinic receptors. That's also true for the somatic motor neuron going to skeletal muscle, and for the input to the adrenal gland. So the first synapse is always the same. The divisions diverge at the second step, the target. The parasympathetic postganglionic neuron releases acetylcholine again, but onto muscarinic receptors this time. The sympathetic postganglionic neuron releases something different, norepinephrine, onto adrenogenic receptors, the alpha and beta types. So acetylcholine and nicotinic everywhere at the ganglion. Then acetylcholine muscarinic for parasympathetic targets versus norepinephrine adrenergic for sympathetic targets. There's one classic exception worth remembering. Parasympathetic fibers going to sweat glands break the pattern and release acetylcholine onto muscarinic receptors instead. 2 ideas that make the autonomic system elegant. First, dual innervation. Most organs receive input from both divisions at once, with opposing effects. The heart gets a sympathetic accelerator and a parasympathetic brake. The push-pull arrangement gives incredibly fine control, like steering with 2 hands. Second, and this is subtle, autonomic tone. Neither division is ever fully off. Both fire continuously at a low baseline rate. So the body doesn't switch a system on from 0. It adjusts by dialing one division up and the other down from that resting hum. The classic example, your resting heart rate is actually held down by ongoing parasympathetic or vagal tone. Cut that tone and your heart speeds up, all on its own. No extra sympathetic push needed. Tone means the baseline itself is an active, adjustable setting, not simply off. The adrenal medulla is a beautiful, special case. Think of it as a modified sympathetic ganglion. Normally, a sympathetic preganglionic fiber synapses onto a postganglionic neuron that reaches an organ. But here, that same preganglionic fiber, using acetylcholine on nicotinic receptors, just like everywhere else, synapses onto the chromaffin cells of the adrenal medulla. These cells are basically postganglionic neurons that never grew an axon. So instead of signaling one organ, they dump their chemicals, mostly epinephrine, some norepinephrine, straight into the bloodstream as hormones. Why does this matter? Because it converts a large local nerve signal into a body-wide broadcast. Every organ with the right receptors gets the message at once, and the effect lasts far longer than direct nerve firing. That's why the adrenaline surge from a scare lingers for minutes after the danger is past. It amplifies and prolongs fight or flight. Let's tie it all together. We started small. Chemical synapses pass signals in a fixed sequence, and calcium entry is the trigger for neurotransmitter release. Those signals arrive as EPSPs and IPSPs, which sum over time and space to decide whether a neuron fires. Then we zoom out. The system splits into central and peripheral, with each brain region owning a specific job. And reflex arcs routing signals to fast responses, directly or through interneurons. And we finished with autonomic control. Sympathetic and parasympathetic divisions that differ in anatomy, transmitters, receptors, and effects, tuned by dual innervation and tone, and amplified body-wide by the adrenal medulla. Signal, structure, autonomic control. That was the thorough line. If you can walk through each of those 6 ideas in your own words, you've got this module. Thanks for watching. I'll see you in the next one. Make sure to subscribe so you don't miss an episode.